Okay, good morning, everyone. Welcome to the Science of Alternative Protein Seminar, the APAC edition. We are super excited that you are joining us today to learn about seafood cell line cultivation. In this seminar series, we host alternative protein researchers in the APAC region to share research advancement and significantly help the alternative protein community identify underexplored technical questions that will help drive the field forward. We hope this series will serve as a platform for exchanging ideas and facilitating collaborations among researchers in alternative proteins. My name is Wasamon, your host for this series. I'm the science tech specialist with the Good Food Institute APAC. We work with scientists from industry, academia, and government bodies to address technical gaps in the alternative protein productions in the APAC region. For today's session, I'm going to start with a quick announcement, and then I will introduce you to our speaker, Dr. Mark Richards. Dr. Richards will then present for the next 40 minutes, and then we will conclude the session with a 20 minutes Q&A period. So before we dive in today, just a quick point on the nomenclature. Some of you may know that at the SIAW, we have announced that GFI APAC have worked with 36 key stakeholders from across Asia Pacific to align on the word cultivated as a preferred nomenclature to describe food products grown directly from animal cells. So while we know that some researchers and companies have used other terms in the past, cultivated is what we will be using throughout this conversation. And I would like to point out a few functionality on the Zoom webinar in case some of you may not be familiar. On the bottom of the screen, you will see the chat and Q&A buttons. We encourage you to use the chat function to introduce yourself and other participants. However, please make sure you direct your questions for Dr. Richards to the Q&A window so we catch all your questions and address them. In addition, in the Q&A section, you have the option to upvote specific questions that you would like to be answered first, so we can prioritize the questions that are most relevant to the audience today. Without further ado, I would like to introduce you to Dr. Mark Richards. Mark is an embryonic stem cell biologist by training. He completed his bachelor degree with honors and Ph.D. at the National University of Singapore. Currently, he leads the life science research team at Nanyang Polytechnic. Mark is well trained in primary cell culture strategies, cell characterization techniques, and animal-free media development. He was responsible for developing the world's first xeno-free human embryonic stem cell lines for regenerative medicine early in his career at the National University of Singapore and ESL International. More recently, he has been working in the cell ag space, developing solutions to address cost and technical hurdles which affect the cultivated meats industry. Mark is a recipient of a Singapore Food Story Theme 2 seed grant for aquatic species, cell line, and low-cost media development. He is also a collaborator in Singapore A-Star Crisp Meat Consortium. Umami Meat is his close industry collaborator. Mark also has research interest in the aquaculture space. Apart from cell ag research, he has funded research projects on mud crab and white shrimp indoor precision and intensive farming system. So we are very excited to have Mark with us today. With that, Mark, feel free to take it away.
Right, so very good morning to everyone. I hope I'm clear enough and you can all hear me well. So I'm Mark Richards from Nanyang Polytechnic, and I'm really very grateful to GFI and Wasamon for the invitation and opportunity to share on my research in the cultivated meat space today. The title of my presentation is Overcoming Some of the Technical and Cost Hurdles of Cultivated Seafood, and I really hope that what I have to share today will be of interest to you. So this is the overview of what I will be covering today. I'll spend just a couple of minutes, elaborating on some of the very exciting developments that we have ongoing in Singapore, particularly in the cultivated meat space. And then I'll go into some of the work that we have done at Nanyang Polytechnic on aquatic species cell line development, low-cost fetal calf serum replacements, and finally, some of the work that we have also accomplished on developing hydrogels and scaffolds for aquatic species cell lines. So this is the Singapore scene today. There's pretty good government support in the form of alternative protein research grants, and we also have a very progressive regulatory framework in the area of novel foods. As you may already know, Singapore is the first country in the world to approve a cultivated meat product. And by this, I refer to Eat Just Good Meat, the chicken from Eat Just. So government support is very good. And we also have a vibrant ecosystem in Singapore, a cultivated meat ecosystem in Singapore. The companies listed here are homegrown Singapore startups in the cultivated meat space. I think there are at least 9 or 10 of them, and at least 4 are focused on aquatic species cultivated meat ideas. So that includes Fisheroo, a new company called Marinus Bio, Shoke Meats, probably the most established of the Singapore cultivated meat companies, and my close collaborator, Umami Meats. So these are some news clippings from local media. Shoke Meats, of course, is the first cultivated meat startup in Singapore, and they specialize in crustacean cells. They've done quite a lot of tasting, and I think they've moved into lobster, crab, and not just shrimp these days. And Good Meat here, it's available and accessible on several platforms in a variety of food products, in a variety of food establishments, from a private members club, and even down to a pop-up they had in a local Singapore hawker center. Hawker culture is quite a thing in Singapore. So a variety of products priced at different levels, available and quite accessible today in Singapore for Good Meat. I took these pictures myself. These are pictures of Good Meat's products, 2 of them, chicken salad and chicken dumpling. I got them on a delivery platform. So, they've even gone into the food delivery platforms in Singapore. Singapore is also home, to the world's first commercial cultivated meat production facility. And, there are many more of these facilities starting up. Esco Ester is also a homegrown company which initiated this, and they also produce quite a few biohazard hoods and clean rooms. Personally, I work very closely with Umami Meats, which is a Singapore-based cultivated meat startup specializing in aquatic species cultivated meat products and platform technology development. Umami Meats has made several breakthroughs in the last year, and they have also organized many tasting events for some of their food products. And finally, Singapore also has a very progressive novel food safety framework. The Singapore Food Story funding platform funds not just projects in terms of in technological, looking at the technology of alternative protein, but they also fund research into the safety of novel proteins. And there's I would say an office called Fresh, which has been tasked to actively look into some of these safety issues. And we have a new guidelines that have been recently issued by the Singapore Food Agency, a new set of requirements for novel food product approval, and there's an abridged version, a set of guidelines that is in place for sensory studies and tastings of these novel food products. So the documents are available at these links. You can submit. They even have a portal dedicated for submission of these dossiers, and they give you a timeline in which you will get a response from them. So I think the Singapore Food Agency should be commended for taking this step forward and being leaders in this area. I never thought it would happen in Singapore, but it has. So coming to what I do, right? My research principally focuses on 3 areas. So the first area is on developing cultivated meat cell lines from high-value and unfarmable aquatic species. And what we are doing here is to look at deriving muscle precursors and cell lines from not just high-value species like grouper, snapper, but also species which cannot be farmed and are still predominantly wild-caught. Unagi is one such example which we have worked in. We also want to work on protected species such as RAS. And I think if you come to Asia, the situation in East Asia is such that we consume really a wide variety and a plethora of different seafood species, quite different from the West, where you probably have salmon and some whitefish. Honestly, if you really don't need to go to Japan, you'd see a huge variety of fish and crustacean species sold at almost every wet market in East Asia. So some of the species which we have worked with and which we are interested in going into include cephalopods. I think they are interesting species. They don't really have a skeletal structure. So it'd be nice to work on these. Protected species, another area, and we have worked on these two, Unagi and a very expensive, high-value grouper species called the panther grouper. In terms of plant-derived calf serum substitutes, the motivations here are, of course, to lower the cost of production. So we are looking at low-cost plant-based serum substitutes as well as plant sources to replace basal media. Particularly, we are interested in the plant 2S albumins and plant transferrins. We have worked on material from legumes as well as yeast, but exploring plant substitutes from fungi and algae as well. And even better, if we can have a media from a byproduct of the food manufacturing industry that can be upcycled into a replacement, media replacement or serum replacement. The third area is essentially that of hydrogel supports for cultivated meat and scaffolds. It's a smaller focus that we have. The motivations are to show proof that we can produce a self-assembling scaffold. We initially started this work not wanting to look at polymeric scaffolds. We wanted a scaffold which could be easily removed from the cells which are growing. So we were looking at self-assembly, small peptide molecules which could self-assemble and form hydrogels. Here we want to look at natural and edible carboxylic acid groups as capping groups. And as I mentioned, a scaffold which could easily be removable. I'm not sure if that would be possible eventually, right? And of course, one of our major drivers here is we were looking at scaffolds which are of non-animal origin. So my collaborators, of course, as I've mentioned and what someone has in her introduction, we work very closely with Umami Meats. We are also part of the Crisp Meats Consortium at the Bioprocessing Technology Institute in Singapore. And I have a close collaborator in the National University of Singapore as well. We are very grateful to Singapore's A*STAR, the Agency for Science, Technology and Research, for funding this project. It represents 18 months of work on this grant, and we have just completed this. So whatever comes next in my slides is the outcome of this Singapore Food Story Team 2 Seed grant, which ran for 18 months. So moving to fish cell lines or piscine cell lines, we have worked with Unagi, and this is probably one of our most ideal and probably the best performing cell lines which we have. Unagi is unfarmable, and this article from the local newspaper Straits Times tells of how the Japanese have tried for many years to complete the life cycle of Unagi in captivity, but have been unsuccessful. So all the Unagi that you get, all the Unagi kabayaki that you get today is essentially wild caught or, juveniles harvested from the wild, put in pens and grown to adult size. In the near future, and we are already, going into this part of our work, we want to look at endangered species and whether or not, several endangered species are actually consumed, endangered aquatic species in this part of the world. There's wrasse, shark's fins, whale, and even sturgeon, right? Moving forward, of course, with the right approvals in place, we would want to look at the endangered species much more closely. So coming to some of the experimental data that we had. This slide summarizes what we have done for Unagi, a Japanese eel. So we procured eels from a Japanese fish supplier in Singapore, and we developed a protocol in which to extract out these muscle progenitors, and also a differentiation protocol to differentiate these progenitors into more mature muscle cells. This is an enlarged image of this section of the picture here, and you can see the striations on some of these Unagi muscle cells that we have. The Unagi cells, in terms of characterization, they do stain for your typical mature muscle markers like myosin heavy chain 1. MF20 is also a type of myosin. One of the issues which we have faced in working with fish cell lines is that a lot of the commercially available antibodies which would work for muscle antigen, muscle proteins in the vertebrate area don't seem to work quite well on fish species. So we went on to further characterize the differentiated Unagi cells. So here we used RNA-seq for transcription profiling, and we compared the transcription profiles of the myoblasts with the Unagi myocytes. And indeed, we can see upregulation of muscle-specific genes upon differentiation. For example, MyoD and myogenin are upregulated in the differentiated Unagi myocytes. So in terms of the protocol which we used, it's standard. It's well, we have adapted it a little, but it generally involves low serum with supplementation of ascorbic acid. Or serum, not entirely essential, but it does improve the yield of differentiated muscle cells at 1 to 2% in horse serum. We wanted to see how far these Unagi myoblasts could retain their differentiation potential into mature myocytes after serial passages, after numerous serial passages. So this diagram essentially shows the Unagi cells at different passages 3 to 12 and differentiated, and staining for myosin. As you can see, after passage 12, there is still a fair amount of myosin staining, but you could also notice that the degree of intensity of myosin tends to drop after every serial population doubling in every serial passage of these Unagi myoblasts. We evaluated different extracellular matrices to see if they could support and promote Unagi myoblast differentiation. The picture over here is that of uncoated dishes. Then we use gelatin, fish collagen, also derived in-house from discarded fish scales, and finally laminin. I think by far, we find that laminin is probably the best extracellular matrix for myoblast differentiation, because the fibers or the more mature muscle cells on the laminin surface are a lot thicker in form and nature. Apart from Unagi, we have also worked on a variety of other tropical piscine or fish species. This is grouper, common tropical species, a very costly, high-value type of grouper as well. They called it the panther grouper and red snapper. So these lines are capable of myocyte differentiation at the early passages, but over extended passage, they tend to lose that phenotype. So some of these lines have also been immortalized, over 200 population doublings without any slowing down in their doubling time. And they're pretty robust and efficient in culture, doubling period about 24 hours or so. The protocol which we use to establish these fish cells, we basically isolated biopsies from juveniles and then subjected these tissue biopsies to both trypsin and collagenase 2 and 4 digest, followed by a pronase digest and filtration through a cell strainer, 40 micron cell strainer. The media we use is L15, And in the initial primary cultures, we did supplement with calf serum and BFGF. The L15 enables us not to rely on CO2 to maintain the pH of the media. So it's actually quite good in that sense. And we can keep the cells at about 24 degrees centigrade. That's the culture temperature. We experimented with both explants and cell suspensions, but I think cell suspensions have a little bit of edge when it comes, or perform a little bit better when it comes to the derivation of these muscle and fin cell lines. So this protocol, we adapted and tweaked a little bit, but it's essentially based on this publication in the Journal of Visualized Experiments over here. So we wanted to see if we could scale up sufficient cell biomass with these fibroblasts, which grow quite well and efficiently. We are lucky in Nanyang Polytechnic to have a Paul Cooperation Center, which has bioreactors for this kind of work. And we use the expansion system from Paul, which is very economical on media usage, to scale up a large number of these fish cells. And we also have capabilities in the school to turn biomass that's been scaled up into prototype food products. So we have a food product development team, a cell line focus as well. As I'm part of that, and we have my colleagues who can develop food products, and we also have a food safety arm in the school as well. So this was done for one of our collaborators. So the next question that we wanted to look at is whether or not the culture conditions that we used and was good and performed well for both freshwater and saltwater bony fish, could these culture conditions be used and be adapted or be immediately transferable to cartilaginous fishes like rays, sharks, and sturgeon, given that the blood of bony fishes is hypoosmotic in relation to salt water, whereas cartilaginous fishes have a different osmoregulatory system. The blood in these fishes is either isoosmotic or hyperosmotic compared to seawater. So we tried this out using explant cultures from cartilaginous fish. These are the placoid scales, which are typical of cartilaginous fish. And while the explant cultures did grow and we could establish primary cells, what happened was that after a period of time, using the same culture conditions that worked for the grouper species, Unagi and the red snapper, we found that the cartilaginous fish cells developed quite a lot of vacuoles. And anyone who's has some knowledge on cell culture will know that this is not a good sign, right? This phenotype obviously shows that the cells are not doing well. They're under some kind of stress and are probably going to undergo senescence soon. So what we did was to adjust the osmolarity of the culture media with sodium chloride, and within a short period of time, the phenotype, this vacuolated phenotype, disappeared from the cells. So we didn't need to use urea, which is sometimes used in attempting to in attempts, right, to culture cartilaginous fish cells. So just adjustment with osmolarity worked quite well. We did not have to alter any other protocols or still reliant essentially on the L15 basal media with adjustment with osmolarity with sodium chloride. So moving on to the second part of my presentation, and that is on plant-derived serum and media substitutes. So here, obviously, the motivation is to get rid of calf serum entirely from the equation. And, it's something which I worked on many years ago in the embryonic stem cell area. And it's something that has come back in big way in the cultivated meat space as well. So we have had success with a variety of pulses and legumes in developing these serum and culture media substitutes. as well as okara, which is a byproduct of soy milk and tofu production. So we have developed a way in which to extract, to make extracts from both legumes and okara, and we have evaluated some of these extracts, about 9 of them, from different plant source materials on a cell line, a robust cell line which we developed as well, a grouper cell line. So what we did was to screen these grouper cells against extracts from various plant species, 9 of them, and we gradually reduced the fetal bovine serum in the media from 10% to 2.5%, replacing it with extracts of various plant species, 2 passages at each reduction and each dilution of the calf serum. We also evaluated subtilisin. We digested the plant material, the subtilisin, to see if that could produce a serum replacement, which would be more efficacious, better in performance. The thinking here is that subtilisin could help liberate some of the smaller peptides, which would otherwise, and increase the bioavailability of general macronutrient content to the cells. So we gradually reduced the serum concentrations, and in total we screened about 9 different plant source materials, mainly from beans and pulses and yeast extracts as well, and 2 plant sources of valorized origin. There's some evidence that subtilisin digestion can improve nutrient bioavailability and increase the performance of these, what we term the plant-derived serum, or PDS performance. And we have 1 or 2 candidates which are performing very well still, and we filed a patent for this invention as well. So what we wanted to do was to see how low we could go, right, in the removal of the calf serum. So again, adaptation, sequential adaptation to increasingly lower concentrations of fetal calf serum. So this is one of our plant extracts, which we term PDS1. In this figure over here, on the left, 10% fetal calf serum control, reduced 2.5 FBS control, and you can see this is quite sparse over here. But with the addition of 7.5% of this plant serum, we see improved growth, and we've pushed this down even further. 1% FBS control, 1% FBS supplemented with 9% of PDS1. Again, there's improvement in growth and more cell numbers here. Addition of ITS does improve and does help speed up the growth, and the increase in cell numbers is quite evident with respect to the control and the test arm over here. Right? So addition of ITS does help, but I think with the 9%, it's quite good, and we're trying to push this down even further to less than 1% FBS. So what we find important in this is that the preparation protocol of these plant serums is important, particularly the dilution factor, because we find that if the plant extract is too concentrated, the cells don't quite like it, right? For some reason, it could be because of the anti-nutrient content or something inside. So the dilution factor is quite important, and it varies from different plant extract to plant extract. Blending of these different plant extracts can improve performance overall, but ultimately as well, cell lines need to be adapted from serum to low serum and possibly even to no serum conditions via clonal selection in the future. Yeast extracts, we got these yeast extracts from a collaborator. So some yeast extracts were supportive, and some were non-supportive. We had filtered and autoclave yeast extracts, lowering the FBS and moving to 7.5% of yeast extracts. We find that they stop performing well, right? At 5% in the previous slide, quite okay, but moving to 7.5%, we see a lot more cell death and a overall decrease in the cell numbers over here. And this project was discontinued, and personally in the lab, we do feel that the plant extracts we have tend to outperform these yeast extracts, which were kindly provided to us for evaluation from a close collaborator. We have a LCMS system in Nanyang Polytechnic using the Shimatsu platform. It's a media analysis package which comes in very handy. It's good to look at how our different plant serums stack up in terms of their nutrient, micronutrient content. And this approach allows us to simultaneously analyze about 150 typical media compounds, which you would find in most medium formulations today. So using this approach, we are able to pick up subtle changes, between basal media and the different plant serums, whether or not certain compounds are upregulated or decrease in concentration. So it's a pretty useful way, a useful approach for analyzing the composition of plant serum media. It's a complex task because there's so many things inside, but this is one way in which we have found, one way in which we feel that it does help quite a bit, right, in making some sense and getting some empirical data to support or reject the performance of a particular plant media formulation. It doesn't detect lipids. Lipids are not inside this particular package. So we did send out some of our better performing plant substitutes for lipid analysis. And on the left of this figure here is essentially the canonical omega-3 fatty acid synthesis pathway. So ALA, or alpha linoleic acid, is a precursor for the well-known omega-3 fatty acids, EPA and DHA. So we sent out, and it's used as a precursor for their EPA and DHA biosynthesis. So we sent out some of our best performing plant serum substitutes. Of course, we did not expect any EPA and DHA in them, but we did find that certain plant media are indeed quite rich in linolenic acid content. And this may augur well for seafood cultivated cell lines in the future, because we do suspect that omega-3 content may be limiting nutritional factor of cell cultivated seafood. So perhaps supplementation with some of this plant media that's rich in alpha linoleic acid could help improve the omega-3 content in cultivated seafood cell lines. So our plant media work so far has focused on whole, right, plant extracts. Moving forward, we want to focus on specific protein isolate fractions to gain a better understanding and a more empirical understanding of what is really important in that extract, which is comprised of a lot of different compounds. So we are looking here at focusing on specific protein isolates from the supportive PDS samples in our previous screens on the grouper cell, on the fish cell lines. And we have developed a low-cost extraction strategy, which gives high yield of plant protein. And we are currently assessing various protein isolates for their culture media performance. And these protein fractions also show quite high solubility, which is an important factor in media development. And in the future, we are paying closer attention to the low molecular weight plant albumin fractions for this part of the work. So screens are ongoing, different protein isolates on our cell lines currently. So apart from albumin, the other serum protein, which is at very high concentrations, is transferrin. And transferrin and albumin are present in serum at probably about 10 to the 3 or 10 to the 5 times higher concentrations than your average growth factor. So can we also derive alternative transferrins from plants or algae? There is some evidence to suggest that there are indeed putative plant and algae transferrin analogs, and this is based on some in silico analysis that's available online, a Markov model. So in this analysis, based on the domain, protein domain and structure analysis, right, homology analysis, it's picked out several plant species, particularly those from beans like chickpea and soybean, which incidentally are also among our best performing sources of media substitutes. And there is evidence to suggest that these beans may indeed contain plant transferrins, right? However, the delivery of iron into cells, transferrin delivery is a receptor-mediated process, which is quite well described. And we're not sure at this point if it would a plant alternative transferrin analog would work on the vertebrate cell because of the receptor-ligand interaction and other considerations. So it's something that we want to look at, and we've drafted up a proposal in which to screen for these plant-derived alternative transferrin or algae alternative transferrins, and submitted this at a recent grant call. So hopefully we'll be able to get funded for this work as well, in addition to the work that we are doing on plant-derived albumins. So moving on to the last part of my presentation, and that is on hydrogels and fish collagen scaffolds. So for our hydrogel work, the motivations are essentially to show proof of developing a hydrogel which can self-assemble, which is preferably also removable from the cell mass, and which is non-toxic, edible in nature. So we looked at small peptide molecules or dipeptide molecules which can self-assemble to give a hydrogel matrix, and this contrasts with polymeric hydrogels, right? We believe that it would be quite difficult to remove the polymeric hydrogel from the cell mass that's growing on it. So we looked at developing proof of concept that small dipeptides of this structure could produce and assemble, self-assemble, right, into hydrogels. So this is what we thought of, right? An intricate balance of hydrogelation. So we, chemist colleague of mine, very good chemist colleague, he's made in the lab a variety of dipeptide molecules which can form hydrogels and which can also transition from the solid state to the liquid state by changing the water content in these gels. So in theory, if we have a cell mass that's growing on a gel like this, and if we increase the water content, the gel would liquefy quite easily. So no heating required, no solvents or any other approaches required. Just increase the water content. And because these gels are non-polymeric, they're made of self-assembled dipeptides like this, they would dissociate and liquefy, thus enabling us to harvest the cell mass and doing away with the hydrogel in the final cell mass that's going to be made into a food product. So he developed a solid phase synthesis method to produce a series of dipeptide molecules. And in terms of the requirements for hydrogelation, the purity of the hydrogel is very important. So these dipeptides need to be of sufficient purity, analytical grade purity. Ionic strength is also important. As I mentioned, if we increase the water content, the gels will dissolve. So we use, but they do form in standard PBS buffer, right? Standard PBS concentration, PBS buffer. Concentration of the hydrogel about 0.4 to 0.7% weight per volume. But one of the major limiting factors, although the gels do perform relatively well, I must say, is that the costs of these dipeptides are actually too high, unfortunately. So the pictures over here at the bottom of the slide show fish cells which have been seeded on a dipeptide hydrogel, 18 hours post-seeding. So they do attach, spread out. We can also incorporate RGD groups into the hydrogels to improve attachment. You may be wondering what the capping groups and R groups, the identity of the capping groups and R groups are, and so I can disclose them. These are the chemical structures of what our capping groups look like and the different R groups, enabling this dipeptide to self-assemble to form a hydrogel. So earlier on, we also wanted to understand whether Piscine cells would grow well on these peptidic hydrogels. So we did a screen on some of these peptide hydrogel formulations and we plated fish cells on it, observed the morphology, and we did use the Resagerin dye, which is essentially a cell viability assay, to understand if cells growing on these gels would indeed proliferate or would they cease to divide. So we do find that compared to controls, plastic, tissue culture, plastic, no gels, we do find that there are some gels which perform quite well, whereas others, essentially, the cells don't quite like them. They stop growing and stop proliferation. But as I said, we put this project on the back burner because of the cost of these dipeptide molecules. For the longest time, we were looking for a lower cost substitute. Actually, the cost factors do drive quite a bit of our R&D in the school. Unable to find any lower cost alternative, unfortunately. So moving on in this space, we are also looking at microbial polysaccharides like gellan gum and xanthan gum. So this is work that's ongoing. I don't have much data to share on this yet, but gellan gum does form a hydrogel at 1% probably, whereas a xanthan gum at 2% might be better to use a lower percentage hydrogel if it can work, rather than something that's of higher concentration, because I don't think we want to ingest too much of this with the with the cells eventually. So we can also incorporate RGD groups. It's work that's ongoing, and I hope to be able to share data on this in the near future. And finally, this is work done by another chemist very good chemist that we have in our school. We're also looking at low-cost approaches to valorize fish skin and here fish scales, and to extract collagen from these products which are discarded from fish processing plants, for example. So he's developed a way in which to extract collagen from fish scales. It's a low-cost method. I believe it's $3 to $4 per gram, and the yield is decent at 11.6% of collagen yield, weight for weight of fish scales. And we've used this as a attachment substrate for some of our fish cells as well. The earlier work I showed you on collagen was essentially collagen from the valorized collagen that's here. But as I mentioned, laminin still seems to be the better performer in terms of producing and giving rise to more mature, thicker muscle fibers. So in summary then, what we have currently is about 10 fish cell lines from food fish, and some of these lines are capable of muscle cell differentiation even after 12 population doublings. 8 lines have undergone spontaneous immortalization, and we've managed to adapt several to low serum conditions with plant extract supplementation. Supportive plant isolates maintain the growth of these fish cells for many passages over 25 and in reduced serum conditions following adaptation. And some plant isolates are rich in omega-3 fatty acids, particularly ALA, and these may be useful downstream for supporting the growth of aquatic species cell lines in order to enrich for omega-3. So we do have several edible hydrogel formulations which support fish cell line attachment and growth, but these peptide precursors are too costly and probably unsuitable for any form of scale-up currently. And we're now evaluating microbial polysaccharides as attachment substrates. Low-cost collagen extraction method at less than 3 grams, Singapore dollars, $3 per gram has been made, and we have filed a patent for some of these serum replacements and are in talks to license some of our cell lines. In the future, this is what we hope to do. I think a lot of effort will be placed on looking at alternative plant and algae-derived albumins and transferrins to further the work on low-cost culture media development, I think ultimately we'd have to look at single-cell cloning and of course suspension culture adaptation, and maybe even fast-growing phenotypes in vitro. Nutritional value and omega-3 content is also on our horizon. And perhaps, we could also leverage ideas from conventional agriculture. There are fast-growing species and stress-tolerant species with the genetics for that. Can these contribute to cell line development? Will, the behavior, will the phenotype in vitro be recapitulated? I'm not sure. What about looking at animal feed and what goes into it? Can we obtain some ideas from animal feed compositions for feedstock development and culture media? And we could also leverage ideas from stem cell regenerative medicine, not just on the technical bits and aspects, but also the safety considerations and aspects too. So with that, thank you very much. I think I'm just about time, and I'd like to acknowledge Dr. Lim Choon Kiat, who was working on this project and who's now joined Umami Meats as the group leader of cell line development. My chemistry colleagues, Chiwi and Subra, very talented chemists, my boss and the director of the school, Joel Mihir, of course, my close friend and collaborator, AP Chan from NUS, and the Singapore Food Story, A-Star, the Good Food Institute, Umami Meats, and the Singapore Food Agency for their participation in this work. So thank you very much. I hope that my sharing was interesting. Thank you.
Thank you so much, Mark. That was a really great and well-rounded presentation. There's so much work that still have to be done, and it seems like you've touched base on many of the aspects of cultivated meat work. So we already have a few questions coming in. So the first question is, will these cell lines that your lab is working and developing be eventually made available for other researchers?
Yes, so we've been approached by the Chris Meats Consortium, which is the group that's focusing on cultivated meat science in Singapore, and they have a cell bank up. We've been approached to ask if we would like to deposit some of these cell lines there. We're still in talks, so I think there may be a possibility, but because there's also some commercial interest in these cell lines, we would have to see, which lines eventually end up in the, I would say, the National Cell Bank that the Chris Meats Institute has initiated. Yeah, so I would say that some would be available. Yes.
Thank you. So the next question is, could you elaborate on how you culture or prepare the cells for culture in 3D suspension versus 2D monolayer?
Yeah. So yeah, that's a good question. So we have not really looked at 3D suspension cultures at this point. So the cells are essentially the hydrogels have been layered on tissue culture plastic. So it's essentially just several microns thick. So I would still consider it's probably not a 3D structure that we have. It's still more or less a 2D monolayer. Yeah, so we layer the hydrogels on tissue on a multi-well dish, and we seed the cells on them. We do see that the cells actually grow into the hydrogel, right? So yeah, but we have not. If the question is about using the hydrogels, attaching cells on them and then putting them in the suspension bioreactor, that's not what we have attempted. The expansion system, which I alluded to from Paul, which we use to scale up the cells, is essentially a multi-stack kind of device. The reactor is a multi-stack device specifically meant for adherent cell culture. Yeah, so we have not done the suspension work in much detail yet. Yeah, hope that helps.
Thank you. So this one question is regarding, I guess, probably a further research. So the question was, the flavor of meat is also influenced by the animal's diet. So and also the enzymatic maturation and aging of meat, which could also alter the texture. Since the cultivated meat process involves growing muscle cell in a sterile and controlled environment, can it give this external factor to simulate a real meat flavor experience?
Yeah, so this is a really interesting question. I don't know really whether or not the flavor of fish cells that which we culture will actually mimic that of real fish, but I think there's a possibility. I hear that from the shrimp people that indeed there is some kind of semblance to real shrimp cells, but I think one way of doing this is to have a at least in a conventional agriculture setting, there's something called a finisher feed, and usually before market, the farmer would feed their animals with this finisher feed, which is enriched sometimes with a lot of oils, to boost the flavor of the final fish product. So perhaps this is something we could do, adapt that idea in a cultivated meat setting, in the final culture condition. We may want to tweak the media to impart a flavor profile, but of course this would entail a lot more work downstream. Yeah, so it's an interesting question, which I think will need to be addressed very, very soon. Yeah, thank you.
Thank you. So the next question is, do cells from your other species exhibit similar differentiation efficiencies?
Sorry, is it the higher fat? Oh, the ALA one? Okay,
yeah.
the bottom one, yeah. Okay, yeah, so it's a great question. We've not really looked at cell line conversion of the ALA. So what I showed was essentially the ALA content in the plant serum, in the plant extract. So I know that so we've not really looked at the omega-3 content in our cells yet. Yeah. So but it's something that I think there are other labs in Singapore who are addressing this question. We have seen presentations from Professor Shigeki's lab. Prof. Shigeki is also in this audience today. So and he's presented some data on this conversion. So I think Prof. Shigeki might be the real person to go to, to get some information on this question. Yeah.
I really like how all the researcher in Singapore is very well connected.
Yeah.
Yes. So the next question is from Jose Burgos. He's a thesis student in cellular agriculture in Chile, and I'm working with zebrafish, and I have about the most
So about the protocol, right?
Cartilaginous and the setting time of the muscle cells in either treated or untreated plates.
Yes. So so that's a great question. It's it's about the protocol. I'll be happy to share some of the protocols we have with Jose. So I have a figure in my mind, but I'm not sure if it's the right one. So I better not say at this point. Yeah, better not say. But I would be more than happy to share some of the protocols with you, Jose. Thanks for the question. Thank you very much. You can contact me. I think I didn't put my email on any of the slides, but you can just look me up. I'm sure you can find me. Yeah, certainly.
So I have this question that I think is very relevant to many people. So the question is on the supplementation of the serum with the plant-based derived serum. So how significant is the cost reduction when cell line is adapted to proliferate in 1% FCS and 9% PDS compared to 10% FCS alone?
How significant is the cost reduction? Right. Yeah. So this is a good question, but we've removed already 90% of the calf serum cost, if let's say if we go with 1% FCS, and we probably can push this down further. I have some figures out there, but off the top of my head, we did do some calculations on cost reduction. I don't have bit off the top of my head right now, but I do have some calculations, and I could share on this, but removing the serum alone would be, I think, would constitute quite a reasonable and substantial cost reduction already. I mean, moving from 10% to 1%, yeah. But we have done some proper calculations, and I would love to share those calculations with you. I don't have them off the top of my head right now. Yeah, thank you.
So I think you've already answered the question from Dr.
Shigeki, yeah.
Cost consideration. So I think
actually the cost, yeah, we have those. It's probably works out to just less than, if I do have it. Just we have some figures, so I better not make the wrong commit to the wrong figure right now, but we do have. It's probably not less than $1 per ml, which some companies are claiming. Some people are claiming, so it's a little bit higher, but it's not. It's still I would say quite a reduction in terms of cost. Yeah.
Yep. Yeah. And I think we're gonna only have time for maybe one last question.
Yeah.
So I think this one is probably quite important. In the cell line development, what kind of phenotype would be the most important to produce the final product for food?
Yeah. Phenotype, as in the composition. Can I just understand cell, looking at specific features which enable more robust growth, or is it or does the question relate to the type, cell type that would go into a food product? Yeah. In cell line development, what kind of phenotypes would be the most important? Okay, so well, I know that at the moment, right? I think at least in the early stages of this field, most of the products will probably be a blend of plant-based protein and cultivated cells. So the form of these products will also probably take on the shape of a patty-like structure. Fibroblasts grow well, very efficiently. They provide sufficient biomass. We need to get a clone, a Myoblast clone, which can efficiently differentiate. We have not got that clone yet, but if we could have all muscle, that would be great as well, right? And but at this point in time, I think fibroblasts give us the cell numbers necessary for tasting sessions, for sensory evaluation. Moving forward, if we could have more muscle, better fat cells, also important for flavor. So I think that's I think it would be that fat and muscle, and with some fibroblasts to make up the majority of the cell mass. So yeah.
Thank you so much, Mark.
Thank you.
So I think that will probably be our last question. For those that we really apologize for the question that hasn't been answered. If you are again interested in to know more, we leave Dr. Mark contacts in the chat box.
Sure. Yeah.
So you can reach out to him. And thank you so much for joining us today. So make sure that if you have other questions for Mark, you can reach him at mark.richards@nyp.edu.sg. Thank you again, Mark, for coming to join us today, and we're happy to have you and learn more about your advance when you do more of this research.
Yeah. Thank you very much for the invitation, and thanks everyone for the very stimulating questions. Thanks.